Assalamu alaikum, dear friends. The topic which I am going to share it is about reproduction in bacteria. So before start the topic, I would like to tell you that what is reproduction. Reproduction in Urdu you can say nasal badana, and the reproduction can be in the form of sexual and asexual reproduction. So let's start the topic, which is about reproduction in bacteria. So there are two methods of reproduction in bacteria: sexual reproduction or asexual reproduction. First of all, we'll discuss about asexual reproduction because it is simple, you can understand it easily. That's why we'll start from asexual reproduction. Asexual reproduction is a kind of binary fission. Here is no any gametogenesis. There is no any formation of gamete. It's just only about binary fission. For example, from one bacteria two different bacteria are formed. What are the steps? The first step is in a DNA. In a bacteria, the DNA combined with the cell membrane, and then it splits into two parts, then replicate themselves and go to its own apart. After that, the cell membrane from the mesosome, the cell membrane separates itself, and then the cell wall also separates, so they become separate and they will be independent bacteria. This is called asexual reproduction. It is very simple. Now let's come to the sexual reproduction. In bacteria there is no any traditional sexual reproduction. There is no any kind of gametogenesis. It is just like another kind of sexual reproduction, and there are three steps, or you can say there are three methods of reading sexual reproduction in bacteria. The first one is conjugation, which is very simple for you. Conjugation, first of all, the steps how the conjugation occur in bacteria. First of all, two bacteria from the same phylum or from the same species, you can say, they came close to each other. They come close to each other, and with the help of pili, they make a conjugating tube. Have you know about pili? Pili is it is just like cilia, but not work as cilia, like a cilia. They combine together and make a conjugating tube, and from that conjugating tube, one of that bacteria donate a part of DNA to the second bacteria. The bacteria which donate the part of DNA is called donor, and the bacteria which receive the part of DNA is called recipient, and the part of DNA is called F factor. Okay, when from the donor part of DNA come to the recipient, so this process is called conjugation. So it comes to the second bacteria and join its DNA. So when the part of DNA join the second DNA, so the DNA is called recombinant DNA. What is recombinant DNA? When two different DNA combine together, they make a recombinant DNA. After this, the second step and third step, the second process and third process will be in the second lecture.